Hi, I'd like to take a little time with you to explain the events of the Crossbridge cycle. This is the series of events responsible for the actual contraction of muscle. Here we see a muscle partially dissected to show a fascicle and one single muscle cell or fiber visible at the far right. A closer look at the muscle fiber shows that it's mostly filled with myofibrils. We see one myofibril extended out to the far right. Looking closer at the myofibril, some structural details are visible. We'll focus on a few of these details. Let me point out the Z-discs. They appear here as zigzag lines in this two-dimensional image, but seen in the real world would look a little bit more like a round waffle turned on its side. These Z-discs mark the boundaries of a sarcomere. A sarcomere is the structure that's actually able to contract. In this image, we see most of the structures of one sarcomere. Again, here and here are our Z-discs marking the boundaries of the sarcomere. Primarily, though, in this image, I'd like to draw your attention to the elements colored red and blue. In this closer view, we see two distinct structures. The red, thick filament made of myosin has lots of golf club-shaped heads. We also see in blue the thin filaments of actin. These two filaments are the parts that actually form our cross bridges. Before we look at the details of how these structures work together, let's zoom in and have a brief look at the cross bridge cycle in action. Notice how the myosin head from the thick filament attaches to the actin on the thin filament, then moves the thin filament toward the center of the sarcomere before detaching. Let's watch that motion again. As you watch the sequence this time, imagine a game of tug-of-war where the myosin head shown is one of many hands pulling on a long actin rope. To begin the cross-bridge cycle, we need what amounts to a start signal. This arrives in the form of nerve stimulation, which leads to the release of calcium from internal stores within the muscle fiber. The increase in calcium concentration within the myofibrils is what allows the cross-bridge cycle to proceed. But how does calcium get the cross-bridge cycle started? Well, at rest, when calcium concentration is low, cross bridges cannot form. This is because tropomyosin, the gold strand seen here, covers the active sites on actin. These active sites are where myosin heads would bind to form cross bridges between the thick and thin filaments. When the calcium concentration is high, the active sites are exposed. This occurs through two intermediate steps. First, calcium binds to troponin, causing troponin to change its shape. Second, the shape change in troponin alters the position of tropomyosin on the thin filament. In this new position, tropomyosin no longer covers the active sites. Now that the active sites are exposed, the cross-bridge cycle begins. We'll go through one complete cross-bridge cycle, which consists of 4 distinct steps. The first step, seen on this slide, is cross-bridge formation. The myosin head binds to actin, forming the cross-bridge between the thick and thin filaments. Besides myosin bound to actin, we also see an ADP and an inorganic phosphate attached to the myosin. These were left by a previous cross-bridge cycle. In step 2, the myosin head pivots using energy that was previously stored. This movement is called the power stroke. The power stroke pulls the actin thin filament toward the center of the sarcomere, shortening the overall length of the sarcomere. As the power stroke occurs, ADP is released, freeing up space right about here for ATP to bind. We begin step 3 as ATP binds. This triggers detachment or breaking of the cross bridge. This is the first of 2 major roles for ATP, cross-bridge detachment. Note that the binding of ATP to myosin is sufficient to break the cross bridge. The ATP itself is unchanged so far. Not until the last step of the cycle is the bound ATP hydrolyzed to ADP and inorganic phosphate. The energy released from this hydrolysis will be used to recock the myosin head, storing energy to be used in the power stroke of the next cross-bridge cycle. At this point, we have completed one cross-bridge cycle. If calcium is still present, as it is right here, the active sites are still exposed, and a new cross-bridge cycle will form, and the cycle will continue. Now, you may wonder, when the myosin head is detached, as it is right here, why the thin filaments don't simply slide back. Remember, we've been focusing on one crossbridge, but there are really thousands of these down the length of any single myofibril. They don't all release at one time. Some crossbridges are releasing while others are still attached. Many crossbridges work together to shorten each sarcomere and ultimately the whole muscle. The crossbridge cycle will continue as long as two factors are present. The first is an elevated concentration of calcium in and around the myofibril. Recall that this rise in calcium levels is a consequence of nerve input. Second, an adequate supply of ATP must be present. ATP is directly responsible for crossbridge detachment, and ultimately, ATP hydrolysis powers sarcomere shortening.